All right. Okay, great. So thank you. Welcome, everybody. And today we're happy to welcome Eva Dyer from Georgia Tech for our seminar. She's a associate professor of biomedical engineering at Georgia Tech, and she focuses on data-centric AI representation learning and AI for science. And so she's going to tell us about large-scale pre-training on neural data to allow for transfer across individuals, tasks, and species. So please take it away, Eva.
Okay, great. Thanks so much for the invitation. And I guess this is a pretty diverse audience, right? So there's folks from many different institutions and from a lot of different backgrounds. So I hope that I can tell you a bit about how we've been leveraging new approaches in AI to, in particular, to provide new insights and advances into large-scale modeling of neural data. So with that, I'll just jump right in. So a lot of the motivation of this work has really come from the immense successes over the past now, now it's going on 5 to 10 years, but we've seen all of these really amazing capabilities that have started to emerge in generative AI, in large language models. And one of the kind of key takeaways, from a lot of these exercises or from this ability is that there's a really important, that scale is very important and can have a lot of power and provide a lot of emergent capabilities within some of these models. So over here on the right, I'm just showing some examples in LLMs where, when you increase the model scale or increase the amount of data that goes into training some of these models, you start to get these sorts of emergent capabilities. So this just shows us that scaling can have a really important impact on our ability to process and make sense of data. This is also something that, Rich Sutton talks a lot about in the bitter lesson. And so, time and time again, we've seen that by embracing scaling and computation, we can often find things within our data that's hard through other kind of handcrafted or small data approaches. And so we were really excited about whether or not this same property or whether or not scaling could be applied within the analysis of neural data, and whether or not it could similarly provide us kind of new capabilities that might not be possible with the current kind of sizes of data sets that we put into our existing models. And so one thing that, is kind of like a good thing. One thing that we can kind of look to is that, okay, so in neuroscience, would we even have the right data, or do we have enough data to potentially explore this possibility? And so the good news is that there has been a really big push, especially over the past decade or so, to develop new tools and technologies that enable more and more neural data to be collected. And so over here, I'm just showing this kind of exponential growth in the size of the number of simultaneously recorded neurons that we're able to capture within a given experiment or a given recording modality. And this is just looking at the growth over time. And what we see is there has been this kind of inflection point as well, where with new tools, we're kind of even scaling up even larger to even bigger populations of neurons. And moreover, in addition to getting larger and larger recordings, we're also now amassing large collections of these data through, great repositories and kind of community efforts. And so, one may then take away from this that we're starting to build up enough data that could potentially, allow us to scale within neuroscience. However, our current models or the way that we go about processing neural data has been really kind of focused on, one model for one data set at a time. And so if we're interested, say, in building a relationship between neural activity and some behavior, or, something about the stimulus or information that we want to decode from that neural population, typically the way that we go about doing this is like, I have a data set, it has some population of neurons, or some, activity profiles that we've collected, and then typically we'll fit a model for each of these data sets one at a time. And so our objective was to, build necessary tools and frameworks that would allow us to start to digest and make sense of and aggregate many data sets into one unified model. So one model that could be trained on many different data sets, that could be from, different individuals or across different days, or, as I'll talk about later, maybe even across different species. So our hypothesis going in is that, neuroscience could likely benefit from this type of large-scale pre-training. So what are the challenges, or what are some of the reasons why this hasn't been done yet? So one of the core challenges is that neural data is very heterogeneous. So unlike maybe genomics, where we might have a kind of standard pipeline or way to collect data, within neuroscience, everyone is interested maybe in describing different functions or different parts of the brain under different sets of tasks. And so, each time a neural data set is collected, it's often focusing on a brain region of interest or using a specific task paradigm to probe neural dynamics. And so this means that these data sets are highly heterogeneous. They're collected from completely different populations of neurons, and while the animal is doing different things. And then in addition, even if you kind of have some of those things a bit standardized, there's a lot of different ways that people go about processing or pre-processing the data, as well as collecting aspects of behavior. And so this heterogeneity has been, a major, well, it's important because we're seeing different aspects of brain function, but in terms of this goal of kind of unifying a lot of data, it's really hard because we're working with highly different data sets. And so in addition to this heterogeneity, if we kind of zoom in a little bit further, like what are the technical challenges that we're facing with? The other key problem is, even if we record from the same brain across many different days or across different sessions of monitoring the brain and its activity, the inevitable nature of the way that neurons are kind of or the way that neurons fire and the way that these technologies go in and access these neural dynamics is across different sessions, even from the same probe, you might find that on one day you capture some subset of neurons, and then maybe on another day you have a completely different set. Maybe you even collect the same set of neurons, but there's no ordering of them, right? Like they're just multivariate time series data sets where the channels or the different neurons that we're recording from are kind of arbitrary in terms of their ordering, right? So that could mean that across 2 days, we look at a data set from the same individual, but the rows of this data matrix have been permuted. And so this lack of identifiability across days basically means that we have to figure out some way of kind of reconciling the fact that we could be measuring from different channels, different neurons. And this has been actually a major challenge for brain-machine interfaces and our ability to, train on some data and then hopefully transfer it so that we could provide new function or capabilities to paralyzed individuals that might want to be able to control different prostheses with their brain activity. So this has been, a major challenge that we have struggled with in the field. And so we set out to develop a framework for training on, many sessions worth of data, and to do it in a way where we could circumvent some of these challenges of the fact that you're recording from different neurons each time. Okay, so how do we do this? So we developed an architecture and training framework that was presented at last year's NeurIPS called POYO. And in the next few slides, I'll go through the solution, the architecture. I know this is virtual, and so it can be a little bit tricky to get feedback, but please do stop me and ask questions or send a chat if there's any part of this that is unclear. Okay, so and I don't know the background of everyone, but I'm gonna assume that folks are somewhat familiar with transformers or the basic self-attention operation, perhaps like in natural language processing. And so I'm going to try to create an analogy to that. So in natural language, we think about, taking words or subwords, right? And from those different pieces of the sequence, which is, a sentence, for instance, we will have a different embedding of each of those words or subwords, right? And so the example that I'm showing over here is, imagine that I have, all these different words within my vocabulary, and then I have some way of forming just an embedding or some vectorized representation of that word. And the idea is that if this is done in such a way where semantically similar words are mapped to similar embeddings, like shown in this case, right? Dog and cat would be kind of close to each other, and maybe pasta is further away within the semantic embedding space, right? So each word has a token that is associated with some d-dimensional feature embedding. And so normally, as we, in natural language, we would string together all of the words in this kind of sequential manner. And that's sort of demonstrated here. So now the question is, how do we tokenize, and how do we take this neural activity data and put it into this same format so that we can leverage transformers or some of the more powerful existing techniques that show us that work well in terms of their scaling capabilities. Okay, so what we do is we will tokenize the neural data on a neuron by neuron basis. So basically, imagine we have 3 neurons that are firing their action potentials, which are shown here in the plot. And so one thing I'll say is, in this first model and framework, we focused on spiking data, or cases where you're recording from neurons and you can see the action potentials or the specific events of electrical activity that are then sent to other neurons, right? And so oftentimes what we do when dealing with this data is we would just take a fixed amount of time or a bin, right? And then count the number of spikes within that bin, and then do this throughout the sequence. So in the end, you take this actually event-based description of the neural activity, and then you turn it into something like a firing rate, so that you can then more easily process the data using these fixed bins. And so instead of doing that, we were interested in whether or not we could work with the raw data in its event-based representation. And so to do that, we basically take each of these events, and instead of binning them and counting, we're instead going to represent each spike, so each activity of all the neurons, as a different token. So that's sort of shown here, where you would have, these 3 or 4 different spikes in this case from the red neuron, each getting a different token for each spike. And so if we work in this kind of event-based framework, then every event from each channel is given a different token, and those are all stacked together in a long sequence. And so So this will be the input to the transformer, and each token consists of both an embedding that describes which neuron it came from, as well as information about the time of that spike. And we can use both of those pieces of information in different ways in the sequel. And so similarly to what I said here about dog and cat being mapped together in this kind of semantic embedding space, we similarly think about these unit embeddings as being a kind of description of the dynamical properties of each of these neurons. So similarly, we might expect that neurons that have similar function or maybe similar cell type to be mapped to similar parts of this embedding space. And this is going to be a learnable embedding that we learn throughout training. So a priori, we don't know which neurons are connected to each other, or or which ones might be similar. And so we're going to learn this over time. Okay, so we're going to have this learnable embedding that will tell us information about the different neurons. What this does is it allows us to now, for a different data set that might not have the same neurons or might have neurons in a different order, we can still learn these embeddings that allow us to sort of implicitly align many neurons over many days. Okay, and so once we go about tokenizing the neural data in this way, the next step is to, be able to then process this tokenized sequential information. And, one way that one could imagine doing this is through a standard self-attention mechanism, so a standard kind of transformer. And so we instead opted for a sort of different approach, which would use cross attention to basically take these sequences, right, of the high dimensional neural activity and basically kind of embed them all into a common latent space. And so what that looks like is basically, so in self-attention, we're computing our, queries, keys, and values over each of the tokens, right? So in that case, I could associate the activity or the embedding of this token with all other tokens in the sequence. And in comparison to that, we're going to, okay, and then what I'll say is that one of the challenges with neural data is because we can have different numbers of neurons, or we can have different amounts of time, or we can have different activity dynamics. So in some cases, they could be firing really densely or high firing, and maybe you have very sparse firing in another. What that means is that these sequences are all going to be potentially really different in terms of their lengths. So in order to kind of mitigate this, and then, as I said, put everything into like a common dimension or latent space, we'll use this cross-attention mechanism. So now the inputs will provide the keys and the values, and then these latent tokens are going to basically query. So you can think about this as these learn latent tokens can learn to query from the neural data in different ways. They could look at the overall firing rate of one neuron, or, maybe they're looking for a kind of correlated activity pattern. And so they can kind of pool in a pretty flexible way and learn many of these different relationships that have to be accounted for. Okay, so then once we get into this latent token space, we then can apply multiple rounds of self-attention in the latents to, build yet more complex feature descriptions and, kind of, get more complex features out. Okay, so we'll apply multiple rounds of self-attention. And then finally at the output, so similarly to how we could treat the input in this kind of continuous way, where we were looking at the timestamp of each of these different events. And if there's more questions about that, I can go into more details, but basically the way that we use that temporal information in the input is through a relative position encoding, where we will modulate our attention scores based upon the difference in timing between the two events. So you get this kind of temporal invariance because you're only looking at the relative difference between the events rather than their absolute position. Okay, so similarly at the output, we can also query. So now we're going to query from our latents in order to produce some sort of output, which in this case that I'll talk about initially is different behavior that's generated during the neural activity. And so what's cool is that instead of having to have like a fixed rate at which you query for the behavior, you can also have this flexibility in your in the time points that you query. And that's just sort of shown here as a schematic where we could imagine producing some irregular output dynamics or behavior. This can be helpful because in many data sets, you might have different sampling rates. There can be mixtures of events that mark trial starts and ends, which are sparse, along with, these kind of regular time series. So it gives a lot of flexibility to be able to work in this kind of irregular way with these data. Okay, so, that kind of provides the framework and the initial architecture for doing this multi-session training. And so we started by testing this idea and understanding our hypothesis, or testing our hypothesis about scale in the context of a motor BCI application. And so here, what I'm showing on the right are all of the different motor tasks that in this case, non-human primates are presented. And, it's worth noting that oftentimes, as I said before, you would only analyze data that's collected from one of these paradigms at a time. And we were the first to consider whether or not, training on multiple tasks could be advantageous. And so in some of these cases, the animal is, pushing a joystick towards these different targets that will appear on the screen here. And then in other cases, they're actually using like a touch screen to be able to move their way around this maze. So really diverse sets of tasks in terms of what we're asking the animal to do. But in the end, all of these things can be described by some underlying kinematics or movement and position of the arm or of the limb. And so our goal is then to train a decoder that can go from neural activity and predict these different movements. So we curated data sets from 4 labs. That's a little bit over 160 sessions worth of data. And, it's just to note that previous models were always trained on a single session worth of data for all these issues that I described before. So like, this is actually a kind of, massive step forward. And these are recordings coming from primary motor cortex as well as premotor cortex across 9 macaques, and contain different types of pre-processing of the data. So when we applied Poyo to this data set, we indeed found really exciting evidence of scaling. And so I'll just point out this one over here. Both of them are looking at if I train this decoding model on a single session and I have something like, I forget how many different dots are here, but each dot is a different session. And so we'll we'll train on a single session, and then we're comparing now to what is the performance that you get with the multi-session trained model. And so one thing that you can see is that, you know, in general, we're always seeing that the multi-session is above the single session. So that's telling us that we're benefiting from scaling. But moreover, when you have more complex tasks like this random target, you see that the boost from multi-session is bigger. And also when you have things that are really hard to decode, you're seeing an even bigger gap. So this is showing us that for small data sets that are hard to extract information from, you can use this multi-session approach to actually, you know, kind of potentially understand or pull out signal from, you know, what would otherwise seem like noise. Just summarizing some of these scaling results. So this is now looking specifically at this harder random target task. And this is the average performance of single session models. So 21 different models in terms of their R squared. Now, what is the performance on those same 21 sessions on like held out trials? So we'll train on, you know, our training split from all these sessions, and then we're testing now on some held out trials. And so we see a big boost in the performance with the unified model. And then what's cool is that now we're looking at what if we, in addition to those same tasks, like those same RT sessions, we now add in 80 sessions from that simpler center outreaching task. And so what we see is that pre-training across tasks also benefits our decoding accuracy. So this is a really cool, exciting result. One thing that I didn't exactly mention, but at the output of our decoder, we also have another learnable embedding for each session. What this allows us to do is then see kind of similarities or have comparisons across different animals or across different labs. And indeed, we do find that there's like clustering of animals from the same laboratory within like just the PCA plot from the session embeddings. So this is, I think, a cool point to make, that we see this type of multi-animal, multitask pre-training as a way of also doing comparative analyses. So seeing how two different animals might process the same task, or how two different tasks might affect the same animal. Okay, so what I've talked about so far are cases where we've trained on data and then we have some test sessions. And so we're asking, how well did we do at predicting on held out on those test sessions? But now we can ask the question of, okay, we have this pre-trained model. Can we transfer it to new sessions or animals and tasks that the model hasn't seen before? And so we developed, or we used, two different fine-tuning strategies on new data sets. The main thing that we have to do is we have to find a unit embedding for all the new neurons that we haven't seen. We have to figure out some way of kind of acclimating them into the model space. So that's one thing. We can just have a little bit of training data on the new animal or on the new sessions, and we can use that just to relearn those unit embeddings, or we can do like a full fine-tuning where we also change the main weights of the model. Okay. And so what we found is that, you know, this also works well. So you can take a pre-trained model and then you can fine tune it in order to do even better. And so here we're just showing the performance on a established benchmark, which is mainly for single session data. And what we find is Poyo, just trained on its own, single session is already, you know, outperforming a lot of the existing baselines. But moreover, we're getting this effect of the pre-training and fine-tuning. We did some analysis to look at like compute and sample efficiency, just that you can do this in like a few shot way. And, you know, you very quickly kind of converge with the pre-trained model. Okay. So, and how long do we have again? I think we have like a full hour, right?
Yeah, although
Okay.
including questions, so.
Okay, so I'll try to do like, yeah. end in 20 minutes then. Okay. So after establishing the capabilities of this type of strategy and showing that, you know, scaling could be a really beneficial thing to do within neuroscience, we wanted to then see, you know, all the different ways in which this could be envisioned and how this could potentially help in other applications. And so in collaboration with Patrick Mignot at Mila and in a joint collaborative effort, we are also working on ways to take these pre-trained models and see if we can transfer to very different tasks. And even in this case, be able to transfer to human data using these like pre-trained models that are pre-trained on macaque or on non-human primates. And so in this example, we we're working on a data set from Frank Willett in 2021, where they basically had a patient who was paralyzed, so actually couldn't make, you know, arm movements. And the goal was to be able to record from their neural activity and then use that to translate into text or into, you know, some intended speech. And so here what they asked them to do is imagine writing each of the different letters, and then our goal was to be able to decode each of the letters in the sequence. And what we found is that, you know, this is the baseline model that was provided with this challenge online. So this is the classification error in this case. And what we found is that if we just do the multi-session pre-training, we're seeing this, you know, major reduction in the error on our downstream task. And then we can go even further if we now pre-train on, or we take the model that I described before, which was trained on 100 sessions worth of non-human primate data, and then we integrate or fine tune on the 10 sessions from human. And so what we see is that indeed there is this positive transfer, even if you're dealing with a very different task and different species. So we're really excited about this potential because it shows us that there are aspects to neural dynamics that are kind of universal, if you will, or could be potentially used to do transfer. Okay. And so in the last part of the talk, I'll discuss our work in now trying to see if we can take this even further and apply to other systems outside of the motor system and predict in this case, so whether or not we can train on diverse data sets spanning different brain areas and even different cell types. And so what I'll talk about is kind of even more heterogeneity in some sense in the data from what I described in the last section. And this is joint work also with Crystal Pan at Mila and my student Mehdi, who led the POYO effort. So yeah, so okay, sorry, I should take a step back. Oh, okay. So where do we go? Okay, so we wanted to find a data set and a sort of set of tasks that would allow us to really test this level of heterogeneity. And so we leveraged this large data set from the Allen Institute for Brain Science, where they have collected calcium imaging recordings. So now it's a slightly different modality. It's regular time series, right? Because you have just these traces that are recorded at regular time intervals. So there's a number of things that are quite different about the setup, but we made minimal modifications to the overall architecture. We had to change the tokenization slightly, but overall everything, you know, is kind of similar in terms of the underlying model, the system. And so here, what I'm showing is now, oh, okay, sorry, I should clarify. Another core aspect of this model that I'll show is, in addition to having, you know, this behavioral sort of sequence that we decode at the output. In dealing with this data set, it was even more diverse in terms of all the different tasks that were being presented to the animals. So we actually trained this, and we developed a multitask decoder that we could use using the rest of the architecture to actually decode many different tasks jointly. So this is trained in a multitask fashion. The different tasks that it's challenged to solve include, so this is the thing that I was missing is like a picture of the data set. So sorry, I'm explaining all this in words. But in this Allen Institute data set, basically the mice are presented with different visual stimuli that have different characteristics. Some of them are simple stimuli, others are kind of more complex, like a natural movie that it's watching. And so some of the tasks that we train this on is to be able to predict aspects of the visual stimuli that they were looking at. So what is the orientation of a drifting grading stimulus? Or in the natural movies case, you know, which frame of the natural movie are you on? Or what natural scene are you looking at? And in addition to all these like visual decoding tasks that can be used to train the model, we also have the running speed of the animal as well as its pupil location. So within this model, we're training on these kind of sparse sequence level tasks, like what is the animal looking at? And then we also have these kind of dense labels that have to do with physiology or behavior. And so we train on all of these tasks jointly. And what we find is that in most cases, we will see some improvement over a model that's trained on many sessions, but only on a single task. There's some cases where we see a bigger improvement of performance, things like the pupil location, you know, increase, or these dense tasks like running speed, both of those actually benefited from training on the other tasks in order to improve performance over just a single task POIO model. But I think what is even cooler about this finding is that due to the nature of the way that these data sets were collected, they have, you know, really diverse properties. So data sets are collected in one of 6 different visual areas. All of them have, you know, really different properties in terms of how they respond to visual stimuli, kind of going up this hierarchical chain of processing. And in addition to these different brain regions, we also have different cell types that we're recording from, and these cell types can be really different in their properties as well. So what we found, which we thought was, you know, pretty exciting, is that in addition to having this improvement in the multitask performance, we also, when visualizing and analyzing the underlying latent representations within the model, we actually found that there was a remarkable ability to start to decode some of these attributes of the neural data. So here I'm showing brain area decoding accuracy across these 6 different brain regions. And this is a baseline model, kind of the best performing model for this task at the time, which is like supervised and uses handcrafted features to be able to solve this region prediction problem. And now I'm just showing, what if we train a POIO, but only on drifting grading stimuli? So it's a single task. And now we're asking, okay, how well can we, oh, sorry, my cat is like getting on my keyboard. Now we ask, how well can we decode the brain region? It is a little bit better than the baseline method, but we see this huge improvement in our ability to start to resolve information about different brain regions from the latent embeddings when we train on many tasks. This is perhaps not surprising in many ways because, you know, different brain regions might be tuned to different types of properties of the visual stimulus. And so we're seeing kind of more more ability to see neural activity patterns that are indicative of these brain regions when we include more tasks. And similarly, we did the same type of analysis on cell types, and so our ability to now decode the Cree line, which is tells us information about which population of cells we're recording from. And again, we find this really nice property where actually it's interesting. So the baseline, which also uses these kind of handcrafted features that have to do with firing rate and the inner spike interval, etc. We had done some prior work on this. So this achieves, 56% accuracy. And we actually find like with a single task trained model, we get a much lower decoding accuracy, which again, might not be surprising because we're not seeing the full range of variability of these cell types in a single task. But we get this, really impressive ability to start to decode Cree lines or cell types with our multitask model. And here's the confusion matrix showing just both inhibitory cell types as well as multiple excitatory types. We also did some analysis of the kind of hierarchical representations or using a hierarchical clustering. And we also found that there was some kind of nice grouping of the different lines that makes sense in some ways based upon what we know. So, so this is a really, I think, compelling evidence that we can use this framework. We can use it in really diverse data sets from different brain regions and different cell types. And it actually gives us this ability to start to see these like comparative differences across the different sessions. We also have been working on other unsupervised methods for learning neuron level representations. So things that would allow us to get closer to definitions of cell type. And so with my student Vinam Aurora, we've been working on, and this is still preliminary, but working on models that allow us to use a contrastive learning mechanism to basically find stable neuron level embeddings by basically taking two views over, two different time points within the neural time series, extracting embeddings of these different views, which will be for different neurons. And then really for a neuron, we want to make sure that its representations are as similar as possible within this embedding space. And all other neurons at different time points can be considered dissimilar or negative examples. And so far, we have seen some, nice properties start to emerge from this type of contrastive learning method. So here I'm just showing the confusion matrix for detecting 3 different cell types, inhibitory cell types. This is a little bit more limited in the cell types we can query or assay because we're dealing now with neuropixels data, so back to electrophysiology. And so in that case, you can only tag one cell type at a time. So again, we find that there's this really nice performance in terms of our cell type classification with this contrastive learning method over the other state-of-the-art methods for cell type prediction. Okay, so with that, I will, I guess, provide a summary of what I've talked about so far. So I hope that I've convinced you that scale is important, and we've seen that this is also true within the neural data sets that that I've described. So with more training data, we can start to extract information that might be otherwise difficult to access. I showed examples of how pre-trained models can be transferred across different animals, across different tasks, and even different species. And also just some early results, as well as, you know, this POYO Plus model that shows us that we can use these types of ideas to extract signatures of cell types and brain region embedded within neural activity. So this is part of a larger effort, both with my lab as well as other groups at Columbia and Mila, to build foundation models or large-scale pre-trained models for neural data. We envision, you know, this neural foundation model that could potentially operate with different types of neural activity. We've already shown how the same architecture and framework could be applied both to electrophysiology and calcium imaging. We have some preliminary results that show that the same model or a model can be trained on both of these sources of data simultaneously due to the flexibility of the architecture. So in addition to different types of neural data, or even potentially language inputs that could tell us interesting information about, you know, the different experiments or the different data sets. We envision, you know, this could be trained across multiple species, as well as many different tasks. And then in terms of the downstream applications that one could envision, I've already talked about, you know, BMI applications. So we're already seeing that pre-training could be a really powerful way to provide much more accurate brain decoders for BMI. I've shown some evidence and examples of doing cell type classification or identification. So, you know, being able to understand properties of different neurons. And we also see that there can just be a lot of ways in which this pre-trained model could be used by others, both for fine-tuning on their own data sets, as well as these sorts of comparative analyses that would allow us to say things about how different animals might process the same task. We think that this can have a big impact on neuroscience. So as I've already sort of alluded to, these comparative inferences across data sets and even across labs, right? So now it's less common for people to analyze their data in the context of other labs that might be collecting similar data. And, you know, this can also provide few shot transfer for BMI, as well as the potential for doing a different type of disease modeling and intervention by having a more holistic picture of what neural activity looks like across the full range of attention and, you know, different tasks and across different animals, different individuals. I think that we will have a better picture of what neural activity, you know, looks like. And this can also allow us to see how different diseases manifest and change those underlying dynamics. And there's, of course, a lot of open questions. There's so much work to be done. I think if we were all in person, maybe I would have gotten some of these questions already. Because like when you see that transfer from monkey to human, right, it seems super surprising. And so one question is, you know, what exactly is being learned during the pre-training that allows for this transfer, especially across different species, right? So it's like, okay, we could maybe think about how there's just kind of there's universal properties for neural activity and firing, and those things are kind of conserved. It's still in the motor area. So even though in the human to monkey case, they're not doing the same tasks or even moving, there's still maybe some common dynamics of circuits within motor cortex that could be shared. And then, you know, how will we now use these types of models to do, you know, understanding of neural systems? What are the unifying principles? And can we use this approach to discover them? And then finally, some data can be more helpful than other data sets. So what are the data sets that we still need, or out of the data sets that exist, which might be the most critical. So this really needs to also be a roadmap for the field to start to think about like how we're going to get to this next step. All right, and with that, I will acknowledge all the amazing folks that made this work possible. So in particular, Mehdi Azabu was the lead on our side, and all these amazing people from my lab that help support. And then this is also, as I said, a collaborative effort with with folks at Mila, so Blake Richards and the rest of the team. And yeah, and that's it. Thank you so much for your attention, and I'm happy to answer any questions.
Thank you very much. That was really cool. Really enjoyed that. Okay, we have time for maybe a couple of questions. Seung Bin, go ahead. I think you're muted. Seung Bin, are you there? Oh, mic is not working. Do you want to type in the chat your comment?
Oh, okay. I see the chats now.
Yeah.
Thank you.
You type your question. I don't know if she can. She can hear. Okay.
Yeah. So
Okay.
feel free if you have a question. Oh, okay.
Oh,
Okay. Got
yes,
it. Got
she's
it.
typing.
Yeah.
Okay. So she'll be typing well. Any other questions? I guess, okay, I'll ask a question. This is not my field at all, but so what is your dream new data? If you would if you could gather the most effective data for testing your framework, what would it be?
It's a great question. And this is something that I've been talking about with experimentalists as well. They're like, oh, if we need to collect data, what do we? So I think it's still unclear what will be the most critical aspect of diversity to kind of control for or to like incorporate into the model. So we have, differences in different brain regions, differences in different tasks, as well as like how much of the brain are we recording from as well. So I think like there's been a big effort in the field to build standardized data sets that are basically using exactly the same task. I mean, we saw the case of the Allen Institute where it is different tasks, but it's all visual, right? And rather than also looking at motor or some of the other senses. And so a lot of that has been working towards this idea of like, oh, we want to control as much of the variability as possible, but just do it at a large scale and then get as much data as possible. And I think that those approaches will be really powerful for training this foundation model. But I think in order to make it like applicable to as many different data sets that are downstream that people are recording from, I also think that there's just a lot of power in seeing the brain in different ways, so under different tasks. And I wonder if, maybe part of this would be to have more diverse ways in which we're probing the brain, so more tasks. And that probably doesn't, it doesn't necessarily answer the question, but I think yeah, there was this move in the field to go from very simplified tasks to very naturalistic tasks, or natural, just like the animals just doing whatever. And I think there's a gap in the middle, right? Where we still need to see brain dynamics under these like different ways in which the animal is moving and behaving. But perhaps we'll need so much data with the full range natural behavior. I'm not sure if that's necessarily the only way that we can get there. So that was a little long-winded, but I think our hope is to start just training on diverse data sets, like not to change the way that people do their science, but to be able to like accumulate all the diverse ways that people do science. So,
Thanks.
okay.
So
Yeah,
we
do
have
you want to read it or
Eli has a question. Actually, there's also Sungbin's question showed up in the chat. So
Okay.
actually, maybe let's do Eli, and then we can go to the chat question.
Yeah. Thanks, Eva, for a really great talk, and nice to see you. Thanks
Good
for
to see you.
coming to our community. The results about Pollo and additional information are really impressive and great. I had a question that is kind of following up on Kate's question and probably related, leading to Sungbin's question in terms of scaling of the data. Did you see that as you add more data, the kind of the accuracy of Pollo improves, and also its generalization to other data sets?
Yeah, so in our initial paper and also in the calcium imaging, we did see the benefits of scale. I think that in the monkey case, there's some aspects of that behavior because it's very low dimensional that perhaps aren't like so complicated, right? And so I think we were starting to see the rate of like increase start to maybe plateau a bit. I'm not sure if we completely got there, but I think part of that could come from the fact that we're using supervised training, right? And so you're just narrowing in on some simpler parts of the data. And then also maybe the task isn't as complex. And so, yeah, while we definitely see signs of scale, I think that it'll probably look really different when we do self-supervised training. And I know you guys have been working on
And that was my
that.
other question is whether these concepts of, it's really impressive that even supervised the training that is focused on particular task can generalize. How do you see ways that self-supervision or kind of unsupervised approaches will be combined with this approach, and maybe preparing the supervision will be more preparing for fine-tuning as you showed,
Right.
fine-tuning
Yeah, I mean, it's interesting. Like, I definitely think that self-supervised training of some form will be essential for really scaling, because otherwise too much has to like match in the data sets to do supervised training, right? So on one hand, I think that self-supervised will be essential to get more and more data into the fold. The question is, what is the right self-supervised training paradigm? And I know you guys have been doing work in, I guess, both like mass modeling and also forecasting, or there's different ways in which one could envision doing this pre-training. I think that it's still not clear what the right approach is, even for time series in general. I think that this isn't quite a solved problem. I would say in some ways forecasting can be quite myopic within time series. And so it's not clear that the same ways that people went about doing it in LLMs will be the right approach for us in time series. I guess that's my main point. I think that there's still a lot of open questions. But yeah, we've been, working on different mass modeling approaches and, kind of these mass autoencoders and getting some nice initial results. But then the other thing that's surprising is like the multitask, it allows you to tap into more data, right? You have to have a new task, but potentially some supervised training could be important for this to like make sure that the model is able to really use the neural data towards the intended decoding task potentially. So I don't know, it'll be interesting like as a field to see where this goes. But yeah, I think probably self-supervised and maybe some combination or of some supervised tasks too. Things like Gato, right, is like multitask, and it, you know, trains on all these different objectives. So
Yeah, thank you. This is indeed sounds like a next research direction. I'll let
yeah.
probably Kate take the microphone and read Sungbean's.
Yeah, there's a
Cool.
question
Thank
from
you so
Sing Bing.
much.
It was written. It was, I wonder if the strategy of tokenization and self-attention was the key that enabled this multitask or even multi-species transfer learning, or if it would be possible to other types of neural networks if you just have a large amount of data sets. I'm not sure if you had a comparison to different algorithms to show this.
Yeah. So there have been other attempts at doing some part of this with like sequential autoencoders or other like stitching efforts with LFADs as well as Zebra. And so in both of those cases, basically what you do is you take all the neurons from whatever recording session, they have some ordering, and then you learn a session specific like MLP, which is meant to take that ordering of neurons and then map them into some latents, and then all of the alignment and other training is done within that. I think that this is inherently going to be really challenging to make scale. And so I think this unit level embedding and like channel specific representation, so like basically separating out all the neurons rather than grouping them together, I think is also an important part of what allows us to potentially find relationships across many data sets. And so, yeah, that's one part of it, is moving away from like a joint representation across all the channels and instead doing this channel level representation learning. I think that's also important. People are also finding that in time series. So things like patch TST or iTransformer, all of these end up working on single channels initially rather than combining them. So I think when you have different data sets that have heterogeneous channels, this can be a really important part of the solution. But then, yeah, I mean, presumably if you have an abstraction, the transformer isn't necessarily like the only way forward. It is an architecture that we know enables scaling, and so it might be the best shot that we have currently. But I think some of these ideas can also be combined with other recurrent networks. Actually, there was a recent paper from Meming and Aisha Vermani where they do this like pre-training on many data sets with like a state space modeling approach, or like RNNs. And there they also show that you can get alignment across many data sets using a kind of meta dynamical systems model, if you will. So I think there's a lot of room for this in other architectures as well.
Great, thanks. Hao.
Hi, Professor. My name is Hao Fan. So I'm a new postdoc joined the University of Washington under the supervision of Ali just asked a question, and also under the supervision of Amy Osborne. Great talk. Yeah, actually we are also working on some of this kind of transformer model to build some of the foundation model to deal with the monkeys for the multi-sector, multi-recording sites problem. So according to this one, I have two questions. The first question that I'm curious for your model, because we tokenize based on the spike time and the spike locations. So I'm just curious, by using this model, did you get a chance to look at the attention matrix to see which part, because when we look at the large language models, we know some of the tokens, they are more connected to each other with larger attention values. I just want to ask, so when you look at this kind of token, what we build, did we find anything that very interested to us? Some kind of spikes, they are more get associated.
Yeah. No, it's a great question. We haven't really gone into much of the interpretability angle. Just through like analyzing the latents later on, as I showed with the kind of looking at Cree lines or brain region prediction. But yeah, we did a little bit of analysis very early on to kind of see how the different latent tokens are like pooling. And it kind of looked like they were pretty localized. Like there were some patterns that were kind of more widespread across the sequence, but a lot of the latent tokens were kind of pooling more from tokens that are kind of within their receptive field, if you will. And so, but yeah, we haven't done too much beyond that. I think it's a really promising direction. But yeah, I think it was like we were also kind of curious to see if we to wait to see if we need to do this before we had like a self-supervised model. But now I feel like, okay, like let's go for it, right? Some interpretability would be really helpful because at the end of the day, we want to understand the brain. So yeah.
Yeah, thanks. So I have another question, which I think is somehow very interesting. So right now we have, in Amy Osborne's lab, so we also collect some of the monkey's data for like a center alteration task and the eye cascade. So we are find another kind of interesting phenomenon. Basically says when we do the recording, we find some of the recording sites, they are just not too related with the behavior. So because I think right now the method that you are proposing, if I understand it correctly, so it's training a supervised way. So I'm just curious if, because when people give you the data, right? When people give you the data, we say because we collect it together, so we say they are just aligned, but essentially maybe some of the neurons, they are just firing in their way and they are just not correct, like related to the behavior decoding. Then what do you think how those kind of data can be, for example, like ruin the algorithm, or it can be like facilitate the algorithm in some kind of implicit way?
Yeah, it's a good question. I mean, I think with the supervised training, the model can easily ignore neurons that aren't related to the behavior. The opposite is often true with, say, neuropixels that we've discovered, where it's like, spanning multiple brain regions. So if you want to decode like a drifting grading stimulus, probably looking at thalamus or V1 is a lot more informative than other regions. So what we find is actually the model will kind of overfit or find specific neurons that it cares about and kind of learn to ignore the rest, which we see as like not a positive attribute. So we do use a lot of dropout to try to encourage the model to still find, the signal from different subsets of neurons. But yeah, I think the other part is doing the multitask training. So I think there were certain brain regions, right, that wouldn't have been strongly firing to drifting gratings. But then when you include all these other tasks, like you start to span the range of all the things that those regions care about. So that's another way forward. But I do think that for self-supervised training, it's going to care about predicting every neuron, right? And so if there's some neurons that kind of don't have a strong predictable signal behind them, you'd have to like learn spontaneous firing, and that might be quite difficult. So yeah, I could imagine how that could be more disadvantageous perhaps with like generative or yeah, like unsupervised models. So
Yeah,
hopefully
okay.
that helps.
Yeah,
Thank
thanks.
you.
Thank you.
All right, do we have more questions? Okay, if not, I would like to thank you, Eva, a lot. Oh, one more, Jingyuan.
Yeah, thanks. I have one question.
You're still muted.
It was echoing though initially.
How about now?
It's just hard. There's an echo.
They are in a meeting room.
Oh, all
what you just
together.
brought all together. Yeah.
I see.
So it might be the ego from the speakers.
Let me try this.
Okay, that's better.
Can you hear me now?
Yeah. Mhm.
Yeah, so thanks for the great talk. I basically have one question about this multitask training. The multitask training, you mentioned like the results of the multitask training showing showing some possibility to identify the neuron like cell types. I was wondering like how this contribute to the cell type identification. Is that because you have more data, or these different tasks really important for you to identify different cell types? Yeah, I want to get more about learn more about this.
Yeah, I mean, I guess it was. Oh, sorry.
sorry.
Maybe you can mute yours because I'm echoing. Okay, there we go. So yeah, I mean, I think it's interesting. I don't think it's because of more data, because when we tried the POYO, which is trained on as many sessions almost, but it was just on the drifting gradings, we actually did worse than like a supervised cell type prediction. So I think that in the multitask, yeah, the inclusion of many different tasks is important because it starts to like you need to be able to understand where the neurons came from or some aspect of their context in order to solve all these different diverse tasks potentially. So certainly with more tasks, we see this marked improvement in the brain region and Cree line decoding. And so I think it's a combination, right? Like you have more data, but probably more diverse tasks to see the neurons under was also an important part of that. So
Yeah, I see. Then the cell type, like how the cell type can be related to the like single neuron identity. Like for example, if that possible to identify the single neuron instead of the cell type.
I see. Yeah, so like in the unsupervised mass audit or the contrastive learning thing I was talking about at the end, each neuron is going to be given its own learnable embedding in the end. And so it might be encoding other properties like, oh, what is this neuron tuned to, or what does it care about, or things that aren't cell type specific. We do see that cell type is kind of, represented in those latents, but there's probably other things that are characteristic and that might be popping out of that. But yeah, there has been some work in the past to show that the neural dynamics of different cell types are distinct in some way. And so the model is finding some way to organize all these sessions in such a way where it kind of ends up using that somehow in its final predictions.
Okay, I see. Thank you. Thank you for
Thank you.
the explanation.
All right, more questions? Okay, if not, then Eva, I'd like to thank you again. That was really great, and we really appreciate it.
Cool. Yeah, thank you so much for the invitation and all the great questions. That was fun.
All right, thanks.